Genetic engineering and bioethics. Huntington's disease. This disease is defined as a hereditary neurodegenerative disorder inherited in an autosomal dominant manner caused by an expansion of CIG repeats in the HTT gene on chromosome 4. This is a genetic brain disorder that slowly damages nerves. This disease mostly affects the striatum, which is a deep brain nucleus that links motor movements to simple tasks. Over time, degeneration occurs in the brain. The overall symptoms take about 30 to 50 years. Early symptoms include slight changes in mood, coordination, and cognition. Mid-symptoms include functional decline, balance difficulty, and motor issues. In the late stages, high motor impairments occur, inability to walk, and trouble speaking. Chromosome inheritance and Huntington's disease. It is a biological staple that every human has 23 pairs of chromosomes. Any error or change to that is most likely the result of a condition or disorder. Huntington's disease comes from a flawed gene in chromosome 4. Huntington's disease is also autosomal dominant, meaning only one copy of the flawed gene needs to be present to inherit this. DNA replication and mutation. DNA replication is division of cells and how they copy DNA. If something goes wrong with this process, it results in mutation. In accordance with Huntington's disease, this mutation is located on chromosome 4. Within the Huntington's disease, the code CAG repeats too many times. The repetition of CAG 35 times is standard and what's considered normal. 40 or more times of repetition is what leads to Huntington's disease. Gene expression in Huntington's disease. Gene expression is the process of converting the information located in a gene to a functional gene product. Gene products are things like RNA, DNA, or a molecule. In relation to genes, Huntington's disease gene is HTT. However, since this disease has too many CIG repeats, it overproduces the protein. Therefore, that protein becomes harmful and slowly begins to damage nerve cells, leading to the original ailment, which is Huntington's disease. Gene regulation in Huntington's disease. Within the protein of gene regulation, the amount of times that gene expression is controlled. In turn, regulating protein, the HTT gene is regulated properly in normal people. This overall contributes to the proper functioning of nerve cells. However, within Huntington's disease, an unregulation for this gene is present. Some can have too many or not enough. Genes need to be properly regulated for overall health. Gene editing proposal. For this genetic disorder, Huntington's disease, I believe the genetic gene technology CRISPR-Cas9 is the best long-term solution for curing this illness. This gene editing tool is very specific. It's defined as the molecular scissors. It has functions to guide the tool in the direction of the faulty gene or area that needs improvement. It can sever genes, correct mutations, insert new DNA, remove bad DNA, and shut down certain genes entirely. Gene editing is a type of technology that lets experts alter or change an organism's DNA. Genes can be added, removed, or altered. In particular, CRISPR-Cas9 can directly remove additional CAG repeats that is caused by the mutated HTT gene, depending on the severity of the Huntington's disease variant in a specific person. The system can be programmed to sever the repeats or reduce the repetition length to an acceptable rate. Some can be born with only one bad HTT gene and other normal. Due to this technology's specificness, the lone gene can be safely removed. Ethical considerations and human impact. To start off, any patient receiving this treatment would slowly start to see improvement of symptoms. Not instantly, however, because the brain needs time to unlearn extensive patterns. There would be improved processing, improved memory, and better impulse control. This treatment also produces different results based on how far along it is treated. Before onset symptoms, serious damage can be avoided altogether. With mid-symptoms, damage can stop worsening, and finally, if it happens to be treated in late stages, progression of this disease can be halted, but impairments still remain. Now, shifting on to ethical impacts, when a gene is edited, all the following generations' genetic makeup are affected as well. This is something that is permanent and cannot be altered in the future. As not conscious or alive, the future generations cannot consent to their genes being altered, even if it was for a necessary purpose, such as removing an ailment or disease. Additionally, like all modern technology, CRISPR-Cas9 is liable to mistakes. One gene being accidentally touched could result in another genetic disorder. Many argue that this method is acceptable for the reason being that it is a way to help and cure those with Huntington's disease or other gene-related ailments. In the past, this kind of treatment was unprecedented. Modern technology, despite its occasional error, has made it possible for some to carry on normal lives. In contrast, some argue that this gene editing ability is unsafe as it is unethical. In the future, some individuals believe that this technology will branch off from health purposes and be used for reckless purposes such as genetic selection and germline engineering. Gene expression monitoring. In order to monitor if the gene editing procedure was successful, further technologies such as DNA microarrays are essential. This form of innovation can be defined as the technology used to determine if DNA from a person contains a mutated gene. The comparison aspect that it utilizes really allows doctors to examine the entirety of a person's RNA within their body. How it works is experts take CRISPR cells and compare them directly with the unedited versions. The two extracted forms of RNA DNA are labeled with dyes to avoid confusion, so they are clearly differentiated. The RNA or edited version of the cells are isolated. Both are then transferred to a microarray slide and viewed closely for inspection and contrast. The absence of DNA can be seen, and it is apparent to the experts that edited cell enhancer still remains the same. If edited CRISPR-Cas9 gene resembles the healthy unedited gene, then the treatment has worked. Signs include an observation of normal amount of CG